Good afternoon, everyone, and welcome to today's webinar, Exploring the Rise of RWE in Regulatory Submissions, sponsored by Cardinal Health Specialty Solutions. I'm Dana Minyard, Director of Technology for HealthEconomics.com. Thank you for taking time to join us in our ongoing webinar series provided by HealthEconomics.com. We're very glad you're here. Before we get started, I'd like to go over a few housekeeping items so that you can participate fully in today's event. If you'd like to resize the presentation screen, you can do so by clicking and dragging in the lower right corner, as shown here. You should see a control panel on your desktop. You can minimize this control panel by clicking the arrow located top left of the panel. You're listening in using your computer's speaker system by default. If you would prefer to join over the phone, just select Phone Call in the audio pane and the dial-in information will be displayed. If you encounter any issues with computer audio, try switching to phone call. If issues persist, most problems can be resolved by logging out of the webinar and then logging back in. Webinars work best when you're engaged. Submit your questions to today's presenters through the questions section of the control panel. You may send in your questions at any time during the presentation. We will collect and address as many as possible, both interspersed through the presentation as well as at the end of the webinar. Engage with us and each other on social media by tweeting or sharing what you're learning in the webinar on LinkedIn or Twitter. You can find us on Twitter at WW_healthecon and at Cardinal Health. A copy of today's presentation is available to download from the Handouts section of your control panel. And now, it's my pleasure to turn today's presentation over to Jonathan Kish. Jonathan, take it away.
Thank you, Dana. Let me just adjust my screen here, and hopefully everyone can see it now. So welcome, everyone. Thank you for joining us this afternoon. It's my great pleasure to be here speaking to you on this important topic. Again, my name is Jonathan Kish. I'm an epidemiologist by training. I've been working in real-world data and real-world evidence for the last 10 years in government and as a consultant for biopharmaceutical manufacturers. Here at Cardinal Health, myself and our team of real-world evidence experts focus on developing that real-world data and evidence to distill the value of novel therapies for stakeholders, including patients, providers, and payers. Today, I'd like to spend one brief moment introducing the colleagues who will join me on this call. Dr. Bruce Feinberg serves as the Vice President of Clinical Affairs and Chief Medical Officer for Cardinal Health Specialty Solutions. Dr. Feinberg works across the Specialty Solutions businesses, consulting with payers, providers, pharmaceutical manufacturers, patients, and policymakers. Since joining Cardinal Health, Dr. Feinberg has made more than 70 peer-reviewed contributions to value-based healthcare literature. And in 2015, Dr. Feinberg was honored as a Pharma Voice 100 Most Inspirational Life Science Leaders. So I'm extremely excited to have him here participating in this panel with us today. I'm also joined by one of our regulatory sciences experts from Cardinal Health. So Todd Phillips is Director of Global Regulatory Affairs for the Regulatory Sciences Group of Cardinal Health Specialty Solutions. Dr. Phillips provides strategic development and regulatory consulting services to a variety of drug and biological product manufacturers. He has more than 15 years of experience in regulatory affairs, drug development, clinical project management, and product marketing. So welcome to both of you, and thanks for attending today and working through this webinar with me. So what areas will we cover today? Well, we've got a lot of ground, as you can see on our agenda. First, I wanted to start by providing an overview of the history, current landscape, and guidance provided by the U.S. FDA regarding the use of real-world data and real-world evidence for regulatory decision-making. We felt to aid in this discussion, it would be extremely important to cover different case studies that show the uses of real-world data and regulatory decision-making. So we will cover 5 cases that have used data in that fashion. They cover both oncologic and non-oncologic conditions, and we hope that they're a good representation of the variety of scenarios in which real-world data and real-world evidence has been put to the regulatory test. So our overarching goal here is to provide you with that background and then work through these examples to show you the highlights and sometimes the lowlights of the use of real-world data in regulatory decision-making. So let's start with our overview. What you'll see on the screen now is a quote from Scott Gottlieb, the former FDA commissioner, regarding real-world data and real-world evidence. As he put it, as the breadth and reliability of real-world evidence increases, so do the opportunities for FDA to make use of this information. That'll be our starting point. There's now a new shift in focus to looking at how we can use real-world data and real-world evidence. But before we get into some of that nuance, let's level set on a couple of definitions. So as promulgated by the FDA, real-world data are data relating to the patient health status or the delivery of healthcare routinely collected from a variety of sources. Real-world evidence is the clinical evidence about the usage and potential benefits or risks of a medical product derived from the analysis of real-world data. So if we're all set on that, we can continue and discuss why this has become so important. Well, in January of this year, Scott Gottlieb also outlined the agency's new framework for dealing with real-world evidence. He said at the time, it's a recognition that new approaches and technologies can help expand the sources of evidence that we can use to make more reliable treatment decisions. And it's a recognition that this evidence base can continue to build and improve throughout the therapeutic life of an FDA-approved drug or medical device. The FDA also mentions that created a framework to explore this a little bit further. The framework is aimed at leveraging information gathered from the patients and the medical community to inform the shape the FDA's decision across our drug and biologics development efforts. This framework will form a cornerstone of our efforts to advance the use of these tools. And in 2019, we'll advance other new initiatives to better leverage real-world data and real-world evidence. We can already see that he's making true on those promises, not only by the guidance and work that FDA is doing right now, but also in his personal life. As many of you have seen, he's now joined the board of directors for Aetion, and as a partner in that, is advancing this science even further. So let's just touch briefly on how we came to this point right now. As we all probably are familiar, real-world data has been used extensively for post-marketing studies. In fact, the impetus largely stems from the early program in 2008 started called the Mini Sentinel Initiative. So there, the FDA program, the Sentinel program, actively monitors FDA-regulated products by EHR, insurance claims data, and registries for safety signals. Fast forward 10 years, and in 2018, there is data on more than 100 million individuals within a network of 18 data partners and collaborating institutions that are participating in the Sentinel initiative. In between the time from the Mini Sentinel to the Sentinel, a few things have happened. The 21st Century Cures Act was enacted by Congress to help accelerate medical product development, and more specifically, it included Section 505F, which specified that the FDA create a framework for evaluating the potential use of real-world data to support approvals of new indications or satisfy post-approval study requirements. As I already mentioned, the strategy and the guidance, the initial guidance on that has been released, and let's cover that in a little more detail. So the key things that the FDA has put out in that guidance relate to three core pillars: the fit for use of the data, the adequate design of the studies that will be conducted, and then the appropriate execution of those studies. Both prospective and retrospective observational studies are considered relevant to effectiveness determinations, and hybrid designs are also approved as well. Fit for use is a very specific pillar for the real-world data, and it really gets to the point of, are the data suitable to address specific regulatory questions? We'll talk a little bit more in a minute about those data considerations. The regulatory question needs to be answered through that study design as well, so that's the middle pillar. And then the appropriate execution is, do we conduct these studies in a rigorous manner that adheres to the best principles of the science that we know? So let's talk a little bit more about the data specifically. So are the data suitable to address specific regulatory questions? Are they fit for use? The two fundamental ideas here that the FDA would like to ensure are covered within the data are their reliability and their relevance. So reliability can relate to things such as ICD-9 codes, Are they specific enough for the condition of interest and available in the appropriate format? Another example would be lab tests. Are they obtained during routine care standardized to remove differences within the organization over time and how those lab tests are measured? Reliability assessments are also a part of this. If the underlying medical concepts are represented by the diagnosis codes and lab values, we want to be sure that that's in fact true. So reliability is really about, are we measuring those right things in the appropriate way? Relevance is, are we also measuring the things that are critical to the ability to make a determination as to the safety or effectiveness of a drug product or a device? So how can we conduct some of these studies? Well, first, it's important to distinguish amongst different kinds of trial designs and studies that will be covered by the program. You can see listed along the bottom there a selection of different approaches that might be used. So while the FDA does not consider clinical trial data as part of a real-world data set or real-world evidence, things such as expanded access protocols, where drugs are given as part of compassionate use or in pre-specified types of protocols, would be considered there. We also have the setting of phase 1 and 2 single-arm trials. In this case, the data that might form the natural history cohort or be used in a comparison could come from a real-world comparator. And then we might have standalone observational studies, which many of us have probably participated in, where there is no trial and there is no comparison in that way. Now, one of the major limitations and concerns that have been brought up in the discussion of using real-world data is that research has shown that many observational studies can't reproduce necessarily or reach the similar conclusions of the randomized clinical trials. So there's a lot of ongoing work being done right now in studying how observational research can be better tailored and designed to mimic the outcomes of those trials to provide the proof of concept. At this point, I'd like to turn it over to Todd Phillips to give a little more background on some of the ways in which real-world data can be collected and used in the regulatory submission process. So Todd?
Very good. Thank you, Jonathan. And welcome to everybody on the webinar. I think this is a great slide. It has a nice pictorial representation which comes from Franklin 2019 that really summarizes 4 use cases in which real-world evidence could be used to support various regulatory decision-making processes. And this would be across the entire development spectrum, both pre and post-approval, and would also include both small molecule as well as biologic products. So in the first case, it's really using RWE to support primary approval or traditional approval of an original application. And so a scenario by which this could actually transpire is if you had a clinical program where the confirmatory trial was based upon or had a single arm within that trial, you could use real-world data to construct an external cohort that could then be used for comparison purposes to the clinical trial data and ultimately determine the efficacy of the product. So the next case scenario is when we're going to use real-world data to support an expansion of a product that had originally been approved under the original application. So in this context, the application is approved by FDA, and then post-approval, the sponsor then could make a decision to pursue an expansion of the indication that was included in the original application. And this could come in the form of expanding the patient population. And so a good example would be using real-world evidence to expand the indication from females to incorporate males as well. And we've seen case studies that have utilized this particular expansion. The next or third scenario that we have there is really specific to accelerated approval. And in this context, you can see where real-world evidence can span both pre-approval and post-approval in the accelerated approval context. And so we know from FDA accelerated approval approvals in that context that for the confirmatory study, they're typically clinical studies. And if the trial shows that the drug actually provides the clinical benefit in the post-approval scenario, then that application is granted traditional approval. So the accelerated approval application is typically based upon a surrogate endpoint. And then in the post-approval environment, the traditional approval is then granted once the clinical trial demonstrates post-approval efficacy. And it could be noted that real-world evidence could be used in this particular setting in the post-approval environment to help FDA grant that traditional approval. And we know that FDA currently right now is looking at the use of real-world data to predict findings of ongoing clinical trials, and those would include phase 4 trials as well, in an effort to develop methods and techniques that would be used to design real-world trials that would be predictive of ongoing clinical trials. So the goal is to have the publication associated with the real-world trial published in advance of completion of the clinical trial. And so depending upon the success that they have in this particular program, which FDA is partnering with a number of third-party providers, I think those learnings from that particular exercise will greatly enhance the ability of use of RWD in the post-approval context, let's say to grant traditional approval for a drug that was originally approved under accelerated approval. And so the next scenario, scenario 4, is one in which the real-world data is used to assess the safety of the product. And that's one that's been used, as Jonathan mentioned, for quite some time, not only by FDA, but other health authorities. And so what we have moving forward in terms of the case studies that we've selected, we will focus on scenarios 1 through 3, and we will not focus on scenario 4 because of the nature in which that data has been utilized for quite some time by FDA. So with that, I will turn it back to Jonathan.
Thanks, Todd. So the remainder of the webinar will be focused on looking at some of these case studies that we've selected. Again, we've tried to create a mix of different types of scenarios, as Todd just mentioned, and also look into those instances where the real-world data and real-world evidence could be seen as a success and aided in the regulatory decision-making process for an approval or an expanded indication, or where we might not necessarily call it a failure, but the new created issues with the data being used in that submission, and so they weren't actually used in that submission. So let's start with the first case study of Brineura for CLN2 Batten disease. So we'll start with a non-oncologic condition, and let's talk a little bit about what this is. I don't know how many folks would be familiar with this disease. However, it's a very, very rare condition. It's a rare neurodegenerative disorder of childhood, actually, characterized by the accumulation of storage material and lysosomes of neural tissues. It has a relatively predictable phenotype with an onset at 2 to 4 years of age, followed by progressive, inexorable neurologic deterioration, resulting in profound neurological deficits by 6 years of age and death in adolescence. So it's a very poor prognosis disease. So the Brineura development program was granted orphan drug designation on April 1st, 2013. A breakthrough therapy designation request was initially denied, and after the submission of additional analyses and data, it was granted in 2016. There were several studies that were used to generate the evidence to support the filing. So study 201 was the phase 1/2 single-arm, open-label, dose escalation, multi-center clinical study. Study 201 you could think of as the standard type of clinical trial that we might imagine for a number of different conditions. What was different about this submission, though, was the inclusion of a natural history cohort to show the progression of the disease and the outcomes, because in this case, such a rare disease in pediatric patients, there are a number of different concerns regarding doing any kind of randomized controlled trial. So study 901 included patients from what was called the DEM-Child Registry, which was an independent consortium that collects and analyzes clinical, genetic, and biomarker data in patients with neuronal ceroid lipofuscinosis, including CLN2 patients. In study 901, this natural history cohort, there were 69 patients who were enrolled. In terms of how the data were collected, you have to think about this as a registry, right? So in some cases, in some cases, patients were followed prospectively as they entered into the registry, but for relatively short periods of time. In some cases, though, obviously the data were fully retrospective, right? So we only have the availability of the things that were recorded in that patient's registry file to understand the outcomes. And what were those specific outcomes that they were interested in measuring? So the primary endpoint was a clinical instrument that was used to collect data on 4 critical domains to the functioning of these patients, of these children. It had to do with motor language, visual acuity, and seizures. And those 4 domains were what was captured and measured in the trial itself, in the phase 1/2 trial. Interestingly enough, then, in the registry, that instrument wasn't fully available. Only the motor and language domains were measured in the historical cohort because the accurate data on those different domains in the language and visual was not available, or it was measured in a different way. So what you can see here on the right-hand side of the screen is the figure from the actual prescribing information for Brineura, showing the comparison between the Brineura-treated patients and the natural history cohort. And what they're measuring here is a standard kind of survival type of analysis, looking at the probability of a decline in those domains that were measured, the 2 domains, over time. The final conclusion of the FDA was that at 96 weeks, in an unadjusted comparison of the 22 patients treated with Brineura, 95% showed no decline in those 2 domains. And that was compared to a decline in those 2 domains of 50% in the natural history cohort. And these children experienced unreversed category decline and really had poor outcomes. So in the end, the FDA used that data, and that data is now part of the actual label and is indicated for the treatment of these patients. So at this point, what we'll do, and we'll do this for each of these case studies, is ask our panelists to weigh in on different portions of it from a regulatory perspective and from a clinical perspective. So my first question for discussion goes to Todd. What were the challenges the manufacturers and regulators faced when comparing data between the trial and the real-world cohort?
Yeah, thanks, Jonathan. Great question. I think in order to put this question into context, I like to think about the endpoints that were used that Jonathan had talked about earlier. And when I think of endpoints, I think of objective endpoints and I think of subjective endpoints. So for objective endpoints, the ones that come to mind are overall survival, hospitalization, and things like that. But then there's also subjective endpoints such as patient-reported outcomes, and in this case, a clinician-reported outcome in which the clinician is providing an assessment of the patient in a subjective fashion that many times requires judgment and things of that nature. And so when you look at the endpoints that were used for approval of Brineura, they definitely used the clinician-reported outcome, which was a subjective outcome, and that ultimately had or transcribed into challenges associated with comparing the single-arm clinical trial to the natural history cohort that was created from the registry data. And so ultimately, when FDA completed their review, they came to the conclusion that the CLN2 scales that were used in both the external cohort as well as the clinical trial were not completely equivalent and had comparability issues. Particularly with the language domain. And so, as Jonathan alluded earlier, the 2 domains that were used were the motor domain and the language domain. But the language domain showed inconsistent ratings that impeded the interpretation and direct comparison between the 2 groups. And so ultimately, FDA considered only the motor domain when they were making their efficacy determination. And so when we looked at the incomparability, the determination was made by the agency based upon a few differences that they noted between the 2 cohorts, that being the external cohort and the clinical cohort. 3 main issues jumped out in my mind. One was the rating methods. that were used in these two cohorts. And so from the clinical study standpoint, the scale was used prospectively. However, in the natural history cohort, the scale was used both prospectively and retrospectively. We also note that the training and assessment guidelines were different between the two cohorts, and the schedule of assessments for when the instrument was administered was different between the clinical study, which was given every 8 weeks, versus the natural history control cohort, where it was given approximately every 12 weeks, and the range associated with that was 2 to 61 months. There was quite a bit of variability on when that scale was given. And so I think this is a great example of when you go to make comparisons between your study and your natural history cohort that was derived from real world data, the endpoints that are used, even in those particular contexts that were the same, the method by which these subjective endpoints or clinical outcome assessments are administered has impact on the comparability of the data. But ultimately, as Jonathan mentioned, particularly focusing on the motor function, the two cohorts were compared formally, and that data is included in section 14 of the package insert, and certainly had impact on the approval of the product. Of note, there's no safety data in the package insert that comes from the natural history cohort, as all of those patients were unexposed to drug at the time. And so this would be a good example, going back to the Franklin slide that I mentioned earlier, of a scenario number one where the use of RWD was in the form of an external cohort. And just as an aside, some factors that make external cohorts more acceptable from a regulatory standpoint there's a few scenarios that are associated with that. One would be promising treatments with large expected effect size. The other one would be treatments for disease with high morbidity or high mortality. And the third and final would be rare disease or a small targeted patient subpopulation. So with that, Jonathan, I'll turn it back over to you.
Yeah, thanks, Todd. Appreciate all the detailed insights there on the study and the outcomes measured. So I'd like to ask Bruce a question then. Considering this indication in this patient population, obviously very small, obviously very orphan, do you consider any of these endpoints differently than perhaps the regulators might? We often think about in oncology, disease response, progression-free survival. How does the clinician and the regulator think about these maybe a little bit differently when it comes to these types of endpoints?
Jonathan and Todd, it's an interesting conversation. I think to a clinician, and I'm going to wear the hat of a clinician throughout, trained as an oncologist for the past 10 years. I've been at Cardinal and have to stretch that hat to be more of an internist, although I'm never a pediatrician. And when I look at data like this, it feels very much like that one-off situation. This is almost a compassionate use design. I think this trial would have been done and would have been favorably looked at by the FDA, irrespective of the 21st Century Cures Act and irrespective of the FDA's determinations on methods for real-world evidence. This is that situation where it's really a story of compassionate use. You have desperate parents, you have desperate physicians, you have a good scientific basis for the treatment, and I think that it's difficult to be able to take this and extrapolate anything from this kind of a case into other cases. When we get into the other 4 cases and we get into the world of cancer and oncology drugs, I think we'll see where the opportunities are to be much, much broader for the applications of real-world evidence, and where the need is also great, but great for different reasons. Not great necessarily just because of unmet need and compassionate use populations, but more because of the significant limitations of randomized controlled trials. And we think of the world of oncology in particular, and it's important to think of oncology not because I'm an oncologist, but of the first 5 cases to use real-world data and real-world evidence, and in seeking FDA approval and achieving approval, 4 have been oncologic drugs. Will be covering them. If we look at the last 5 years of the FDA new drug approvals, oncology drugs have had a very significant representation. Oncology clinical trials have been plagued by very small % population accrual. Current numbers are estimated to be 3% of adults are participating in clinical trials, and those who are participating are not representative of the population of patients who are being treated. They're often, or almost always, healthier, younger, less diverse, more health literate, better social support, and the list goes on. And if we add on to that this rapid expansion of precision medicine and looking at populations, even with diseases as common as lung cancer, to be broken down into subpopulations that are orphan or ultra orphan based on a molecular profile, a gene profile, then we start to see where there is going to be tremendous need in very common diseases to start to look at ways that we can apply real-world data and real-world evidence. So in this particular case, I think that it was one where compassionate use really was a driver, and I think the FDA would have been forgiving on design. I think as we start to move forward, we're going to see where that litmus test is not going to be so simple, and we're going to see the range in which the manufacturers have gone through much more extensive and creative designs to achieve the goals.
Great, thanks, Bruce. And so let's jump into those next examples to keep the discussion moving forward. Appreciate those insights. So our next study is in regards to Lutathera for the treatment of neuroendocrine tumors. So gastroenteropancreatic neuroendocrine tumors, or GEP-NETS, are relatively rare. They're complex neoplasms that present many clinical challenges. One of the key things in the assessment and treatment of these GEP-NETs is the location and extent of the tumor. Traditionally, they've been divided into three different locations: the foregut, including the esophagus, stomach, proximal duodenum, liver, and pancreas; the midgut; and then the hindgut. Survival with patients with GEP-NETs depends upon that stage in histology. Patients with well and moderately differentiated distant metastases have a 5-year survival probability of 35%. However, those with poorly differentiated distant metastases, that 5-year survival drops to about 4%. For the management of these patients, traditionally included surgery, radiation, cytotoxic chemotherapies, and then more recently, the somatostatin analogs and novel biological agents. So Lutathera is actually a radio-labeled somatostatin analog that has a long-running development program. Two studies, or I should say 2 trials, were conducted, and we'll talk about those in detail now. So the primary efficacy of Lutathera was studied in the NETTER-1 trial, which was a randomized, multi-center, open-label, actively controlled trial of 229 patients with somatostatin receptor-positive mid-gut carcinoid tumors. That was the primary trial. In addition to the NETTER-1 trial, in the protocol for that, there was an expanded access program that was written into that protocol. So that's the second trial referenced below, the Erasmus study. So the Erasmus study was that compassionate use expanded access protocol, which allowed patients with the neuroendocrine tumors at different types of sites. So again, thinking about the foregut, midgut, and hindgut, et cetera, to be receiving Lutathera. In that long-term expanded access protocol, over 1,214 patients eventually received Lutathera for those GEP-NETs. It's interesting to note that this Erasmus protocol was, and the analysis plan for that was written 8 years after the initial study initiation. There was no specific sample size or hypothesis testing plan that was discussed in that specific protocol. So what did they do with this Erasmus data and the NETTER-1 trial specifically? Well, when it comes down to it, in the end, both of these data sets were considered in the indication for the approval of Lutathera. The primary source was the NETTER-1, and you can see the table showing the outcomes for those patients treated with Lutathera in the NETTER-1 trial. And then you can see also the control arm for the long-acting octreotide. However, what is different in this case is that the NETTER-1 trial had a very specific definition of the locations of those GEP-NETs, and the Erasmus trial was actually expanded beyond those specific locations. So amongst those 1,214 patients who were treated in the expanded access program, they were actually able to go back and find 360 patients who specifically had GEP-NETs at those external locations, who also had RECIST-based evaluations conducted, and extract that information to demonstrate the efficacy of Lutathera, not only in the NETTER-1 trial population, but in a slightly expanded trial population as well. So going over to Todd then, and we'll again do this kind of as we go through, but how did the patient data collected under the expanded access protocol inform the FDA approval in this original application for Lutathera?
Great. Jonathan, thanks for the question. And going back to the Franklin side that I reviewed earlier, this would be in scenario 1, where the real world data and evidence was included in the original application that was reviewed and ultimately approved by FDA. But typically when we think about real world data from an actual product, I usually think of post-approval collection of that data. But this is a good example of using an expanded access protocol during the initial development program to collect RWD on the product in a patient population. So we know in the expanded access programs, or as Jonathan mentioned, the compassionate use, this pathway is typically used to grant access to investigational drugs when patients have exhausted all approved treatments and can't participate in the clinical trials. And we know that these particular protocols are likely to collect data on a more heterogeneous population than would normally be included in the clinical program. And in fact, we've seen that with the Erasmus data and the location of the GEP-NET. So I think this is a great example of data collected during the initial development program from an RWD standpoint that absolutely had an impact on the approval of the application in terms of expanding the indication statement. And we see the use of the RWD and RWD within the package insert in the clinical trial sections, as well as in the safety section. So it's a great example of collecting RWD during the initial program and utilizing that particular data for approval of in this case, broader patient population for the initial indication.
Thanks, Todd. And then over to Bruce. So Bruce, building upon our conversation from a few moments ago for Brineura, how do you believe in this case the similarities or the differences in the patient populations might have impacted that FDA decision? And how do you see the expansion from just the clinical trial to this real world cohort? And are there things we should be concerned with there, or are they just very similar when it comes to the clinical nature of the disease?
Thanks, Jonathan. So for those who are less familiar with what we've been talking about, you may be familiar with the term carcinoid, and this collection of tumors has often been referred to over the years as carcinoid tumors. And it is the simple fact that we have these different terms. We're dealing with a tumor type that has still been historically defined by light microscopy rather than by its molecular and genetic profile. And to some degree, these definitions are holdovers of kind of an older era in the way we thought about cancer. And so we're watching almost before our eyes as we look at the study, the movement from organ-based disease to cellular-based disease, and today to molecular and genetic-based disease, which is often tissue of origin agnostic. And in this case, that study supports it in a way because we're looking at somatostatin receptor positive tumors. And to some degree, you could argue that it could be irrespective of location. And I think that's all flavoring the era that we're in and study design that began 10 years ago versus FDA decision making now. And so the historical differentiation has been between pulmonary NETs, neuroendocrine tumors, and GI, and that relates to carcinoid syndrome and the way in which the disease manifests itself symptomatically, and to some degree, that separation of foregut, midgut, hindgut could be argued as arbitrary. But nonetheless, although these although it has been in part of different staging methodologies, which continue to refine and reform literally every decade for the last 5 decades. There's been another reevaluation and presentation of a new way to consider NET tumors. All of that, I think, has to flavor when the FDA is going to look at this. So it's a little bit different than maybe a tumor type or a disease situation, which is somewhat more perceived as more static and well-defined. And I think in that case, there's somewhat artificial separation of foregut, midgut, and hindgut when you really have a more molecular definition at hand, which is somatostatin receptor positivity, played a lot into the willingness of the FDA to look at the real-world evidence as they did. A second important feature here is the fact that most of these patients, I shouldn't say most, but a great number of these patients, because of the lack of effective therapies, do enter into clinical trials and are referred to academic institutions. And therefore, when they are not in clinical trials, they may be managed in a department or in a clinical setting in which specialists who care only for this type of tumor are seeing them, and the radiologists who are involved in the imaging of these patients are also only looking at cancer and may only be looking at a very restricted group of cancers. Very different than what we see in the community and the real world evidence we may be collecting from the community. So the fact there were 300 plus patients who had in the real world evidence arm that had RECIST-based assessments on their imaging is something that is very different than what you would find in looking at real world evidence in the community, where the RECIST is unlikely to be used in most community-based hospital radiology departments. And even if it were to be used by radiologists, it wouldn't necessarily be formally recorded in the radiology report and is not part of that community physician's assessment of that patient, which is often much more of a gestalt, which takes into account the imaging, which is likely a narrative, much improved, as well as biologic and performance-based factors, weight gain, improved labs, improved quality of life, decreased symptoms. So I think it's a very interesting study because it brings us into the world of cancer, but it also has some interesting differences from what we'll see in other studies.
Great, thanks, Bruce. I think you made a great point there about the reliability, right, and that the 360 of those patients had the RECIST criteria applied. So let's move ahead, and for the audience, just to let you know, I know we've got 3 more case studies to get through. We're going to hold questions and try to get through these as quickly as possible, hopefully with time for questions around 1:50 p.m. or so. So our third case study is in relation to Palbociclib, so Ibrance. So as many of you all know, Ibrance is approved in combination with letrozole or fulvestrant for the treatment of metastatic breast cancer. Interesting here is the idea that Pfizer sought to expand the indication into female breast cancer. So to achieve that expanded indication in a very rare tumor when it comes to male breast cancer with an incidence of only around 2,670 cases a year, where conducting a study might be unfeasible or unethical, they sought to design a study to use real-world data to support demonstrating the safety and perhaps the effectiveness of Palbociclib in those patients. So where did the data for male breast cancer patients treated with Palbociclib come from? Well, there were 3 data sources that have been discussed, and I'll preface this by saying that all of the typical FDA reviews and commentaries are not as available as of yet to showcase how all this was thought of by the FDA compared to some of the other studies we've already talked to. We've gleaned this from a lot of different sources, but essentially there were 3 databases: the IQVIA insurance database, the Flatiron breast cancer database, and the Pfizer global safety database. So you can see here on the screen how they kind of looked at different things, right? So the safety signals we can obviously understand how those might have been collected. They also did look for tumor response, and that was pulled from the electronic health record as far as we understand it, looking at several different factors, including descriptions of the radiology findings. So I think important here, and we don't necessarily know the full extent of this, but to Bruce's earlier point, the levels of evidence when it comes to using RECIST-based imaging studies and evaluating the tumor lesions versus what was described in the clinical narrative for these patients, I think weighs in how the FDA might consider some of this different evidence. In the end, based on these data, the safety profile for men treated with Ibrance was consistent, and so the FDA did approve the supplemental NDA to expand the indication to men. So a question to Todd then. The FDA had previously extrapolated efficacy and safety data by granting indications in breast cancer to male patients, even though male patients were enrolled. So did the Palbociclib real-world data support this extension of the indication to men, and how was the safety and efficacy data used used differently if it was?
Great, thanks, Jonathan. And I think this example is a good case study for a post-approval extension of the initial indication approved by FDA, when the initial indication, of course, was in women. And the question becomes, what impact did the RWD have on extrapolating the efficacy from women into men? And so my personal opinion, based upon the data that I've seen, is FDA certainly used the safety and efficacy of Ibrance that was demonstrated in women in this decision, but they also most certainly used the real-world data that Jonathan had just mentioned in the extension of this indication. So as the question alludes, FDA has definitely previously extrapolated efficacy and safety in breast cancer indications to males, even if no males were enrolled in those particular development programs. However, there are certain cases where there's a potential of different efficacy and safety results between men and women in breast cancer, such as when a drug is combined with an endocrine therapy and results in or relies upon the manipulation of the hormonal axis, which we see here. And in those cases, additional data may be needed to allow for the extrapolation into men. I definitely think this is one of those situations where the RWD that was provided by Pfizer gave the additional information that gave FDA the comfort in the context of an endocrine therapy and hormonal access to expand that indication from women into men. In terms of how the data actually appear in the product labeling, there are reference to the electronic records within section 6 of the labeling, which is the safety section, in terms of the post-approval exposure of that particular product in men, with a reference to the similar comparability to females and males in a post-approval standpoint. And that's it. So information used to support the extrapolation is silent, but safety information as it references males and females is present in section 6.
Thanks, Todd. So over to Bruce for a quick question here. So is this extrapolation, I mean, we can all probably think that it's probably clinically appropriate. Was this one of those cases of kind of a slam dunk, as we might say, for the expansion of this indication? Or how would you describe this, and what other nuances do you see that might apply to other oncology therapies?
So this definitely, in my mind, fits into the slam dunk. And for both the Ibrance story and for the GEP-NETS story, it's interesting because you almost could argue why go through the process if you're the manufacturer. Certainly in the year in which the studies were designed, we were ICD-9 code based. The distinction is not made for the location of a neuroendocrine tumor that's GI. And in many cases, you could always use a not otherwise specified code determinant. You could do the same thing in terms of breast cancer, and obviously Ibrance was being used in men who were hormone-sensitive breast cancer in the metastatic setting, because that's where they developed the real-world evidence patient pool from. And so there is that question of even why do the study. So kudos in a way to the manufacturers for doing the studies, and the beauty of real-world evidence design, especially when we're looking at label extension, is that it removes all doubt with a relatively low-cost approach to the problem, as opposed to launching another clinical trial that would cost millions, if not tens or more millions of dollars, and take years. These are studies that can be conducted in less than a year, relatively low cost, and be able to not just assert that historically males were treated exactly the same as females and had the same clinical benefit, but rather be able to put proof behind that. So I think it's an interesting story. Again, kudos to manufacturers who are doing that. But there are all those questions in terms of even original design, to be the fly in the room, the why behind it.
Great. Thanks, Bruce. So we've only got several more minutes, and we'll try to get through these, I think, as quickly as possible. So let's talk quickly about Avelumab for Merkel cell carcinoma. And I think even if we don't get through all the next study, this addresses some of the insights into when real-world data kind of falters a little bit. We've seen 3 great examples of successes, so let's talk a little bit about where it falters. So Merkel cell is obviously an ultra-orphan aggressive skin cancer with a very poor prognosis. The study of Avelumab, the Javelin Merkel 200, was an open-label, single-arm phase 2 study of patients with histologically confirmed Merkel cell carcinoma. A total of 88 patients were followed for a median of 29 months, and the primary endpoint was durable overall response rate. The sponsors wished to use data from a retrospective chart review to support the submission. And so what they did was use the iKnowMed EHR from McKesson to identify Merkel cell patients to establish that natural history. So when they went into that database, they found 686 patients who were confirmed to have Merkel cell. And I think the interesting thing here is of those 686, once they applied all the criteria from the trial to get down to that final patient population, they found 39 patients who could be potentially eligible for study. Of those 39, it was boiled down to a much smaller cohort of actually, in the end, 14 patients for whom disease response could be assessed and the objective response rate reported. So here we've got a case of trying to use a very large, robust database, but in the end finding very few patients who could be used in there. And I would add here that I forgot to mention that the study of Avelumab was based on the second-line treatment of patients with Merkel cell, so following progression after first-line chemotherapy. So let's just talk briefly about the results and then get into the questions. But in the results of the Javelin trial, the objective response rate, or ORR, was 33%. In the retrospective cohort, again, of 14 patients, it was 28.6%. So the FDA has approved Avelumab for the treatment of metastatic Merkel cell. The prescribing information only contains the results of the Javelin study, and there's not a reference to the real-world data specifically for the efficacy. And interesting enough, the FDA decision is such that it's not just for patients who have progressed after first-line chemotherapy, but Avelumab is indicated for the first-line treatment as well, and includes patients who are 12 years of age and older. So to Todd, what relevant factors and data did the FDA consider to scientifically justify extrapolation of the efficacy beyond the patient population included in the Javelin study? As I mentioned, the study was in second line. The study didn't include pediatric patients. So how did that happen?
Yeah, great. Thanks, Jonathan. And so the question I have in my mind, too, is this similar to Ibrance, where the RWD, in my mind, definitely played a role in the extrapolation of the efficacy? And I think in this scenario, the answer is no. And so FDA extrapolated into not only chemotherapy naive patients, but also pediatric patients using different methodology that didn't involve RWD. So for the expansion into the chemotherapy naive patients, they looked at various factors such as ongoing clinical trials with this particular product, as well as the unmet need for metastatic Merkel cell, and in addition to the favorable toxicity profile. When it comes to pediatric patients, they extended that down to adolescents to the age of 12, and that was supported by a population PK analysis. So in the end, I don't believe RWD played a role in the extensions that we saw away from the clinical trial, the Javelin study.
Thanks, Todd. So over to Bruce for a little commentary here on the endpoints, perhaps, or other thoughts on this study. When evaluating disease response as a surrogate endpoint for PFS or OS, are there limitations to the use of real-world data? I think you've touched on some of those, but maybe just expand a little bit on your thinking here in terms of Merkel cell and how we might assess the efficacy or effectiveness of therapies using real-world data.
Yeah, so it's a good example, as you start to move down and go through an attrition table of EMR for eligible patients, and you go from hundreds to tens, and even with that, you're still looking for where any of those patients formally resist. In the case of a Merkel cell, is resist really applicable because many of these patients have bone scans and PET scans that are being added to their CT? How is that really going to be done? And it raises some real serious concerns going forward, as it looks like real-world data and real-world evidence has a very significant role to play. But what's going to be the design of the studies that are going to be done either as a real-world control arm cohort in an approval study or for a label extension study? And I think it raises a serious problem. And there's a lot of research being done, and it's good quality research looking at surrogate factors using claims like time to next treatment and treatment duration, or in the case of trying to use image reports plus what was charted regarding best response and using RECIST type criteria, but only in terms of it being a PR, CR, and ORR, not in terms of the basis for that measurement. And I think it's very difficult. And the critical determinants will be in the next couple of years. Can there be new and creative methodologies to try to overcome those differences to truly be able to do RECIST, Lugano, Chesson, and match it up directly against the randomized controlled trial work? We've been experimenting with that. We've gotten to publish a little bit of our work in that field, in which we're asking physicians to directly abstract the image because images are available to most clinicians through PACS. Those are portals that hospitals provide for imaging data, and make those measurements and provide those measurements rather than going just from a report, which may or may not have the data in there. So I think there are some interesting and creative ways to try to approach this, but to me, that's going to become the holy grail of being able to advance real-world evidence and data into the next era.
Thanks, Bruce. So I know for the audience, we only have a few minutes left. We may stay a couple minutes extra if that's okay, and we understand that you can't. And for this last case study, I will probably hold off on questions for Bruce and Todd and just walk through it so you get the information you need, and we can open it up to any of the questions that came through on the audience. So just to discuss this briefly, the case here is for Selinexor for multiple myeloma. So Selinexor is a novel treatment that was evaluated in the STORM trial, a phase 2b study, in combination with dexamethasone for the treatment of triple refractory multiple myeloma. The primary study endpoint was objective response. In the submission to the FDA, they included data, the manufacturer included data from a retrospective observational study using EHR data from the Flatiron database. And the hopes of that was to characterize the survival distribution of those patients in that real-world cohort and compare that to the actual STORM trial when it comes to those types of data. So again, in the interest of time, I'll jump ahead. But there were several conclusions and several ongoing things here that happened. Initially, when the FDA reviewed that data and has provided this feedback in their ODAC briefing documents, they identified several critical issues to trying to use that real-world data in comparison. First and foremost, the analyses were not pre-specified, and there was no discussion of the protocol and other analyses plans with the FDA. Second was a selection bias type of issue, where the populations enrolled in STORM couldn't be appropriately matched to those who are coming from the real-world database. And so at that time, based off of that, the FDA concluded that the evidence generated for the real-world data analysis in combination with the STORM trial data was not sufficient to grant that approval. They recommended awaiting further results of an ongoing phase 3 trial called the Boston trial. And that was back in, I think, roughly April of this year. At this time now, though, however, the good news would be that this drug has been approved for the treatment of patients who are refractory to 4 different types of agents. And that approval was based off of the manufacturer submitting additional data from that Boston trial, actually, which showed the benefit of Selinexor. So in this case, the real-world data was not successful in helping to establish the efficacy or the safety of Selinexor in these patients. So we can maybe talk about these. Let me just, we've got 1 minute left. We'll stay for a couple extra, but I have some questions here for the audience, and I'll just start with one that might go to Todd. What is your advice around study design discussion with the FDA prior to study implementation or even during the study? So maybe you could speak to that briefly.
Yeah, in terms of engaging the agency, and I think it's dependent upon how you intend to use the RWD. But if you intend to use it to support the primary approval of your product, as we've seen in some of the earlier cases, earlier interaction with the FDA is recommended. So in the scenario where you've got plans to conduct a phase 2 trial that could be a single arm trial or could potentially have an internal control arm within that particular trial, but there's also the option of potentially having a single arm trial in connection with a real world cohort that you could use instead of having an active control cohort, let's say for an example, in your clinical trial. In that context, and this is also assuming that you intend to submit the application for marketing approval based upon the results of those phase 2 trial. In that scenario, I would definitely recommend having a discussion with FDA to discuss how you intend to execute that real world study in support of an external control arm, particularly the protocol and the SAP. I would definitely have those submitted to the FDA and have dialogue around those 2 particular documents in advance of executing that trial.
Thanks, Todd. And with our last minute here, I'll kick it over to Bruce for one kind of final question and closing thoughts. So Bruce, maybe you could summarize your feelings here on the use of real world data, and specifically, do you see the FDA in your mind, or even from the perspective of the clinicians, how do you see them evaluating or adjudicated the reliability and the evidence? Will this continue to be on a case-by-case basis? Is it simply too difficult to try to recommend blanket kind of recommendations, or how do you see that moving forward?
So I think there'll be more consistency as time goes on. This is just as much an experiment for the FDA as it is for the manufacturers who are going to pursue this direction. And I think over time, they will develop a more refined response. And as we saw from our presentations, they were a little bit all over the place in terms of when they were critical, when they were less critical, and the differences between the nature of the disease and the urgency of the treatment, the availability of competitors. There were all those factors that seemed to be weighing in that decision making. It wasn't strictly a quality of evidence story. So I think it is going to change over time. I think it behooves all of us who are involved in the field to be thinking about how can we do it better? Where are the gaps and the unmet need? What can we do differently? How do we get that data? And in some cases, there are limitations. If you're in the EMR business, more structured fields, we're going to have to pursue things that are outside of the normal reach. If you're an oncologist and you think there is an opportunity, you have to recognize that if we don't change the radiology community's perspective, if they don't start adopting, completing all analyses and reports that are with the disease-specific imaging criteria that are being used in the clinical trials, and is that even reasonable? If it's not reasonable, to what extent can AI be used in order to create, machine algorithms that can do that, as certain ones have already been developed, but not for these specifications. So I think there's, again, a huge opportunity. I think we're in a growth phase now. I think there's learning as we go, but I would believe that it's not going to be long, maybe 2 years, before we really see a very consistent approach to how the evidence will be viewed.
Great. Thank you, Bruce. And let me thank all of you, first our panelists, Todd and Bruce, again, for participating today. Thank you all for staying a few extra moments. If there are specific questions that we didn't get to, please don't hesitate to reach out to me at this email address, and I can forward on the question to the appropriate expert as needed. And with that, we'll close. I'll turn it back over to Dana at healtheconomics.com. Thank you.
Thanks very much, Jonathan, and thank you everyone for attending today's webinar, Exploring the Rise of RWE in Regulatory Submissions, sponsored by Cardinal Health Specialty Solutions. Remember, if you have any questions about the webinar or would like to follow up with any of today's speakers, email Jonathan at the email address you see on your screen. Once you leave today's webinar, you'll receive a survey on the presentation. We would really appreciate it if you would complete that and provide your feedback. You'll also receive a follow-up email shortly with a link to view a recording of today's webinar. On behalf of Cardinal Health, healtheconomics.com, and today's panelists, thank you for joining us and have a great rest of your day.